Hi, everybody. I'm Bill Bengston, president of the SSE, and although it's probably already happened for you, welcome to our crazy conference. Hope you've had a good day so far. I think this is being played Thursday night, so I'm hoping you're going to have a good night also. I'd like to thank a couple of folks and move on because there'll be thanks going on during the conference. First of all, in terms of the actual physical layout and how this stuff works, we've got Healthy Seminars with Lauren Brown, Lorianne, who's running this and actually recording this for me. They have an extraordinary team, and without them, we would not be able to pull this off as a Zoom conference. So thank you to Healthy Seminars, Lauren and company. And obviously, the reason we're here is John Kruth and company. John is the head of the program committee, and he's put together a wonderful lineup. I hope you're having fun. I hope I'm having fun, and we have a good weekend. So to John Kruth and the entire program committee also, thank you very much. So I'm going to give my presidential address, try to keep it as brief as possible. And really, this address is a continuation of last year's. It's not a prerequisite to get in. But last year, I was talking about transitioning from the way we used to be to the way we ought to be. And in one sense or another, I'm doing the same thing here. Last year, when I gave this address, I was really talking about a couple of things, a couple of themes. I think that we've spent, and this is SSE people, and I think also PA people, and people who study anomalies, whether you're in the societies or not, I think we've spent too much time dealing with or dialoguing too seriously with people who consider themselves to be skeptics. I don't find a whole lot of skepticism out there. I'm a skeptic, but I don't find a whole lot of skeptics out there. I find believers who come in various stripes, various flavors. So there are people who believe everything that people at conferences like this say is true. There's people who believe everything that people say at conferences like this is wrong. And very few people actually know the data. And I think we spend too much time dialoguing with believers, and I don't think believers are ever seriously convinced by data. And I spent a little bit of time last year, and I think you can look at this if you want to go online to the SSE YouTube channel. I spent a little bit of time last year suggesting that it's an interesting question why people convert from this position to that position. Why is it, for example, that someone accepts, and let's go out of anomalies, why is it that someone accepts that the COVID vaccine is safe? So right now we're in COVID land. So that the COVID vaccine is safe and it's a good thing to do. And there are people who adamantly believe that that's true, and there are people who adamantly believe that that's not true. And yet there's a database there that I wonder how many people are looking at it. So this wouldn't be considered anomalous, but I'm raising the question, why is it that people accept what they accept, and why is it that they believe what they believe, and what gets people to go from position A to position B? And I suggest to you, just as a quick comment, that data is rarely the case. And I'm going to bring that up sometime during this talk. But I would suggest if you're trying to dialogue with a believer and you think you can convince them by showing them some data, you're going to drive yourself nuts. And it's not, we spent too much time dealing with believers. Let's deal with skeptics. Skeptics being people who are curious, open-minded, and don't know for sure. When I look at anomalies, I tend to be curious. I think, to be open-minded, and I don't know for sure. Even about my own stuff drives me crazy. If we look at a variety of anomalies, let's go to we talk about the COVID vaccine as a conventional idea. Let's go to a non-conventional idea. Let's go to the PAIR lab. Well, the PAIR lab, as I think everybody here knows, spent almost 30 years showing, among other things, that consciousness can affect random number generators. Now, they did this for almost 30 years. And at the end of 30 years of extraordinary experiments, zillions of experiments, an enormous amount of data, I wonder how many people have been convinced, who are convinced that something like this is impossible. And I would recommend to you People aren't going to just say, oh, look at this. We have 29 or 20, 28 years of data piled up, and therefore that's enough. I don't think that the Pear Lab, which I consider to be a rousing success, would have been more successful had they done an extra year of data. I don't think there's anything you could say to a believer, if they're believing on the other side, to get them to look at, take seriously, or spend the time examining the data to see that, in fact, something serious is going on here. And so I'm I'm trying to look at the idea of when we're out there dialoguing in the world, let's look at things that are doable. An extra year of the Pear Lab doesn't make it better. An extra decimal point in an experimental p-value is not going to make it better. We have an enormous amount of data. For those people who are data-oriented, beautiful. For those people who are believers, they don't care. Now, I'm going to talk today about turning the unconventional into the conventional, which is another way of saying essentially the same thing. And I'm going to focus here on some healing experiments. Can we capture healing intention and reproduce it without the healer? That's really the question. So let's get trucking through this. And the bottom line, in case you want to leave early, if you're going to make healing conventional, and right now it's not, it needs to be two things, and remember these two things from my entire talk. It has to be storable, and it has to be scalable. If we have a system that's storable but not scalable, we're probably not going to transition to conventional application. If we have something that's scalable, but it's not particularly storable, the same thing will happen. We're not going to have this turn into a conventional type of therapy. And I'm looking at healing and trying to take it mainstream. So we need mainstream storable, we need healing mainstream scalable. So we look at images of healers, and you can get the idea here. Now, this is just an image I took off Spotify, nothing more exotic than that. And here we allegedly, at least Spotify is allegedly, looking at someone who's a healer and someone who's a healee. And for reasons that make no sense to me, the healee here has his eyes closed. I have no idea why his eyes are closed. I have no idea if this is supposed to be some spiritual thing that's going on or whatever it may be. But nonetheless, we have a healer, we have a healee. We have one person on one person. This is the idea of healing that most people have in their heads. The problem here is that you have an inexhaustible supply of people in pain. You have an inexhaustible supply of people who need healing. And we don't have an inexhaustible supply of healers. And so let's say this healer does something to the healee and the healee gets better. Well, that's beautiful for the healee. What about the rest of the world who needs healing? That's what I'm looking for. So again, the two main pillars here are it needs to be storable, it needs to be scalable. Now, we're used to this all over the place in the world. We just have never really thought about it seriously as it pertains to healing, or actually a number of other anomalous phenomena. So we can take static electricity, we can take charges, we can do all this stuff, and we can store it. And so everybody here, I don't need to ask, is familiar with a battery. And what have we taken? We've taken a natural phenomenon out there in nature and we put it into a box and we've stored it in the box and we can hook stuff up to that box and take the charge out of it. That's beautiful. So charges which exist in the natural world, we can put in and make an artificial device, put the charge in there and call upon it later. That's beautiful. So if this is a storable system for charge, and it is, it doesn't necessarily mean it's scalable. That's scalable. So we take a battery, we take a bunch of batteries. We're not taking a battery, we're not making a single battery, we're making off the assembly line a boatload of batteries. If the boatload of batteries have the charge, have a usable charge, and can be mass produced, we have storable, scalable. And we know this not just in the physical world, obviously, we know this in the biological world. And so we're used to vitamins, we're used to supplements, we're used to medicines that we take. And so we go to a physician, conventional, and they give you a prescription, conventional, and you go there and you get a bunch of pills from a pharmacist. Well, that means that the healing or the augmentation of the healing is storable and it's scalable. If I've got pills here, they look like fish oil pills. If they're fish oil pills, we can take fish oil, we can store it, and we can mass produce it. It becomes conventional. And we could go on and on with those kinds of examples. So here I'm looking at, can we now take this phenomenon of healing and turn it into something which is storable, scalable, and conventional. So the plot, how I'm going to get there, a couple of minutes on some previous healing studies. This will be quick. And then we're going to look at some experiments indicating that healing can be stored, and it can be stored in organic material, and it can be stored in inorganic material. I'm going to look at some experiments that show that healing intention can be recorded. Now think about this. If we have a recording, we have scalability. If we have a recording, the only question out there is not scalability, it's storability. We're going to look at some experiments looking at water. Turns out water is a wonderful medium within which you can store healing intention, but that doesn't make it scalable. So the question is, how do we combine this, and how do we put this together to turn this thoughts on going forward, turn this into something a little bit more conventional? So that's where we're going. That's the plot of what I'm trying to be talking about here. So I've been doing the research for, I think, 105 years, and I've done conventional kinds of research in healing. So I've looked at the parameters of healing. I've looked at a whole bunch of correlates of healing, physiological correlates, psychological correlates, on and on. The third bullet, development of a theoretical model, I stink at. I mean, I just stink. I don't, after doing this 105 years, I don't have a good working theoretical model that would allow me to make specific predictions. I just don't have it, nor do I know anybody else who has it. But the good news is, even if we don't understand something, we can use it. So what I'm suggesting is basically going from, let's balance what we're doing. Basic science is a beautiful thing. Let's also talk in terms of an applied science. So can we make practical stuff? And that in healing means we can capture it, we can reproduce it, we can scale it. So I draw on a boatload of experiments, a big boatload of experiments. And probably the ones that I've done that most people know about is my cancer experiments with mice. And I've done a lot of cancer experiments with mice. These are formal experiments, City University, 2 experiments, St. Joseph's, 2 experiments, Arizona State, 1 experiment, 3 at Indiana Med School, 5 at Brown University Med School. This is going to be 6 and 7 in a second. University of Connecticut, 2, and we go on and on. These are formal experiments in vivo with mice checking out cancer stuff. And can healing interfere with cancer? I've looked at oncogenic mice at UC San Diego. I looked at nude mice back at Brown University, aggressive mice at Wake Forest, and I just finished a couple of experiments at Tokyo University on breast cancer, bladder cancer, and melanomas. So there's a lot, and the stuff is ongoing. And what I do when I do my experiments is I think in very simple terms. I think in terms of, what do we think we already know? So I start with, if you will, conventional, and then I introduce something unconventional into the system. That's the general plot. So in the case of mice, for example, what really matters in here, we're taking a mammary cancer, one of the forms I've experimented on, and you inject the mice with a certain amount of cancer, details to follow, and the mice live no more than a month. And they really don't live more than a month. So you inject them with cancer, they die an ugly death. No mouse ever makes it past a month. And there's a boatload of experiments. Guesstimations are a little over 2,000 experiments on these mice. So there's enormous amounts of stuff known in the conventional biological world. And what I do is I interject a single variable. What happens if we take a model that's already known and we interject healing into the middle of it? That's really the question. So we're going, if you will, conventional. I throw in an unconventional. How does that change what we already know about the conventional? So we take mice. This is a mouse about to die. It's an example of this is an external tumor. It's non-metastatic. This is the cancerous tumor. You can see the fur is falling out. If you can see the eyes, there's nobody home. This is essentially a dead mouse. It's at the very, very, very end of its life. And it will die, and it will die within the next couple of days. No mouse ever makes it past a month. And I interject a single variable. So this is a picture of me, because I don't want to out anybody else. I use faculty and students and such as healers, but I'm not going to out them here in this presentation. So this is me, and this isn't the COVID garb. This is, you have to be like this in a lab. So this is before COVID. You had to wear all sorts of stuff because we were dealing with immunocompromised mice, nude mice. And so we have a mouse that's going to die. We interject healing, and then we go back and we find out, knowing what we know, what should have happened, what actually happens. And what happens is this tumor gets a slit, opens up, ulcerates, and then implodes. And the mouse is not made better, the mouse is cured. And I use the word deliberately, the mouse is cured. It's cured for its entire life. And more than that, if we re-inject it with cancer, no cancer will take. Now that's reasonably interesting. So again, we start conventionally. We got a mouse model done by all over the world. We interject a single variable and we see what happens. That's what I do. And when I do that, just really quickly, we have cures all over the place. And we have a whole bunch of correlates to this. Distance, it turns out, is irrelevant. Dose response is real. Healing is not a field effect. And we could go into other stuff if this were a different talk. Belief of the healee is irrelevant. And actually, it might work against. Now, to be obvious, mice don't believe, as far as I can tell, at least they do, they won't tell me. Cell cultures don't believe. I got a lot more studies than this. But people, for reasons that still don't make sense to me, think, I must believe, or if I believe, this is a good thing. And it turns out I've tested this, and belief is irrelevant. Turns out it doesn't matter whether the healer or the healee believes. A non-believing, a skeptical healer and a skeptical healee is about the most perfect healing setting you can come up with. Whether you're dealing with plants, whether you're dealing with mice, whether you're dealing with people, whether you're dealing with cell cultures. So belief, we find, or I find, is irrelevant and actually might work against it in the case of humans. I've gone into a bunch of stuff on placebos we're not going to talk about here, but I get placebo effects in mice that are exactly the same as people. I get placebo effects in cell cultures. I get them all over the place. I don't think placebo effects are psychological. I think they're an actual case of physical resonant bonding. Whoops. Healing is not PK. Healing, I think, instead is stimulated by the biological need of the healee. And I think the most important factor in the healer-healee dynamic is the healee. And I think that the healing is actually generated by the healee. Again, it's a separate talk. At the same time, healer and healee go into a resonance. I've measured brains using EEGs and functional MRIs, but you get the idea. So there's a boatload of healing out there. Notice all of these questions are basic questions. So if you're a traditional scientist, you say, can you cure stuff? Does distance matter? Does dose matter? Is it a field effect? And there's all these questions that you raise, and they're all good questions. What I'm suggesting is we've got the answer to a lot of these questions. Let's move on. Let's not reinvent the wheel. Let's not reinvent the past. Let's not keep going. Let's not say, if I just do one more experiment, I'm going to convince a believer whose belief is that this stuff is not right. And so we can put this a slightly different way and ask what matters most. And I would recommend to you that the folks at this conference, including me, are stuck to some extent, and I don't mean that in a negative way, but are stuck on the first two bullets. I care about a theoretical explanation, even though I don't have one, and I deeply care about the quality of data. And I'm not suggesting here that by any means that we ought not to be concerned about theory or that we ought not to be concerned about the quality of data. But I'm saying that basic scientists, basic researchers focus on the first two bullets, and normal people, as opposed to us, normal people tend to focus on the last two bullets. So a regular person on the street says, well, that's kind of cool. Can I experience a healing? Can I experience being the healer? Can I experience? Can I experience? And the other thing that a regular person will say on the outside is, what can you use it for? Cute, but now what can we use it for? So I think we've spent a good amount of time on the first two bullets. That's a good thing. We have not spent enough time on the second two bullets, and I'm going to focus here on the last bullet. So if this stuff can become conventional, again, it needs to be stored, it needs to be scaled. So I'm going to show you a couple of experiments, or just talk about a couple of experiments pretty quickly. And I'm going to look at organic storage of healing. And by organic and inorganic, I'm not talking about the way it's used by most people out there, organic, inorganic, using fertilizer, all that stuff. Do you have free-range chickens? I'm not talking about that. I'm talking about, is it carbon-based? That's all I mean. So I mean, if it's organic, it's carbon-based. If it's inorganic, it's not carbon-based. I'm not talking about pesticides. So let's look at organic storage of healing, cotton. So I have a prop here. So if you can see me in any little corner there, I'm holding up a piece of cotton. What happens if I treat this cotton as if it were a mouse? And I put my hands around and I do a healing technique and I do my stuff and I have the cotton. Can this cotton, now I'm calling it loosely charged cotton, can this charged cotton reproduce the healing? And so the thing is pretty simple. We get already a known model. We've got cancer cells in a well plate. Think Petri dish. We've got cancer cells in a well plate, and then we take the cancer cells and we bring the cancer cells closer and closer, and we put them on top of the charged cotton. And we take cancer cells and we put it on charge of cotton that has not been charged. And what happens? Well, really quickly, if the cotton's been charged, there are genomic changes in the cancer cells. And that's reasonably interesting. And so if the cotton's not charged, and you put the cancer cells on top of uncharged cotton, cancer cells look at you and go, what? What do you want from me? If the cotton is charged, the cancer cells genomically change. So I don't think the cotton believes. I don't think the cancer cells believe. I think we have a natural phenomenon here. And again, we have a known model. We know what's supposed to happen. We introduce healing, but now stored healing, and we find out what happens. Other organic storage, cell medium. So this is the stuff that is used in labs in particular for in vitro work. So you've got cancer cells in a well plate. You got to have a nourishment. You need an environment that they can thrive. And off the shelf, you get this stuff, and this becomes the soup within which the cells live. So it's the nutrient bath that the cells in vitro live in. What happens if you treat not the cells, but you treat the medium that the cells are stored in? So now, instead of a piece of cotton, you're thinking you're holding a bottle of these cells and you're mailing them, you're holding a bottle of these nutrients and you're mailing it to a lab, and the lab then puts the cells in them and we see what happens. Now what happens, and this was UC San Diego, what happens is pretty dramatic. It's beyond a statistical effect, clearly. And here's the real short version of what happened. The cancer cells, when exposed to treated medium, grow wildly. Average growth rate of treated medium cancer cells is 600% increase in division. Now, what that means is the cancer cells, and this horrifies some people, the cancer cells who people hang out, the cells are going to hang out in, people think cancer is a bad thing. If cancer is in your well plate, if cancer is in your Petri dish, if cancer is in your test tube, if you got stored cancer, and that is your healee, that's your client, what should happen to cancer? Well, cancer is the client. What does cancer like to do? It likes to reproduce. Healing is allowing the cancer to do what it likes to do. If you're treating a mouse, the mouse needs to fix the mouse. If you're treating cancer cells, healing is helping the cancer. I think, haven't done this yet, that if we took charged up cancer cells and put them into a mouse, they would collapse, but that's a speculation. I haven't done that yet. So human leukemia cells grown in treated medium will explode in the amount of reproduction that it does. Organic storage. So we got it in medium, we got it in cotton, and we can do this actually in mice themselves. Now this is a bad picture. I've gotten off Spotify. It's not really a mouse, it's a rat. It's a better picture. But you get the idea. So on the left, this is hypothetical, on the left, we have a treated mouse. Someone's come along, put their hands around, done the healing technique. And on the right, we have an untreated mouse. And obviously we're not going to have one and one. It's going to have a bunch here and a bunch there. And we take blood from a treated mouse and we inject it into an untreated mouse. Everybody gets cured. Now that's reasonably interesting. So the untreated mouse that's never been exposed to healing hands-on has been exposed only to a mouse that has had hands-on work. And we're taking blood from that hands-on worked mouse and putting it into a non-hands-on worked mouse, and we'll cure that mouse. So there's memory, if you will, there's biological memory in the organism itself. Now, where else could it be stored organically? And the answer is, I don't know. Think of all the things that are possible. But we've put it in cotton, we've put it in cell medium, we put it in mice, we've put it in a bunch of stuff, and we find out healing intention can be stored. Doesn't mean it can be scaled, it means it can be stored, at least as it regards organic material. Now, it also can be done with inorganic material. So I'm going to do water very quickly now, part one. I'll be back with part two. And in water, which is inorganic, even if it has no pesticides, we're taking treated water bottles. These would be stuff you'd put in mice cages, and we treat the water bottles and never meet the mice. And the treated mice, treated only with treated water, will be cured. So just as a quick, we know it should have happened, we know it should have happened, we know it should have happened, and sometimes what should have happened is an organic should have happened, sometimes what should have happened is an inorganic. So again, we've got storage, doesn't mean we have scalability. So let's change the question now to scalability and look at some experiments that try to articulate scalability. And the way to maximize scalability, if we were looking for a method, would be really simple, a recording. If we can actually record using whatever method of recording we have, and a recording can be stored digitally in the cloud, we could make this scalable to the extent of around the globe in about a minute. So if we have storage by a recording, we've got global scalability. If we've got global scalability, it assumes that we've got storage. We can have storage without scalability. We can have scalability without storage. So I'm starting here with first maximum scalability. What happens if we try to record healing intention? So I have a team that has come together. You know some of these folks, at least some of the SSEs. Everybody here is a member of the SSE. Certainly the folks in the PA recognize Dean. So we got Dean Radin, John McMichael, Mike Turner, Sarah Besum, all SSE members. And they've got the skill set that I needed to exploit in order to test whether healing can be recorded. So at IONS, this is Dean's Faraday cage. We got a big wonkin' Faraday cage blocking out hopefully as much interference as we can. And inside this Faraday cage, we put a bunch of stuff. And so we basically have a lot of detectors. We're not sure exactly what we're doing, but we're loading the place up with detectors, and we're running healing as we're running the detectors. And the method was going back to cotton. We've already demonstrated that cotton can store. And we put 3 people in this Faraday cage holding a piece of cotton and charging the piece of cotton while the detectors all ran. That's the general idea. So at the end of this, it's reduced to a WAV file. It's a recording. The recording can be shipped. The recording can be uploaded. The recording can be used like any other recording can be recorded. So we've got the signal recordings, we've got the recording, and then we try to see, does the recording reproduce what we were getting in terms of healing? Again, if it does reproduce, we've got global scalability. So here we have a setup. This is at Brown University. We have your basic amplifier, your basic speakers, nothing exotic, and we have human cancer cells down here, and we play the recording through the amp into the speakers, and then the cells, I'm using this loosely, listen, they hear it. And then we find out what happens. And we know what's going to happen if you put cancer cells in an incubator at a particular heat level and you run them, you know what's going to happen. You know what's going to change or not change. And so we introduced the single variable, but now it's not a person. Now the single variable is a recording of healers healing a piece of cotton. And we looked at time sequences, and we looked at 167 genes, and we can reduce the 167 genes to stuff that's of particular interest to us. And they have to do with telomere length, increase, upregulation of apoptosis, upregulation of immune function. Something serious happened. And it happened seriously to 68 genes, and we did this 7 times. So we have a reliable effect, replicated, which turns into a reasonably interesting paper in a conventional biology journal. Now remember, if I'm in a conventional biology journal, it may be an unconventional thing, but it's sufficiently interesting even to a conventional biologist. They say, this might be something might be going on here. How does it work? I don't know, but something's going on here. So the results are not trivial. 7 replications, 68 genes change out of 167, and it's basically the same 68 genes which change if exposed to this recording. So in this particular paper, we have 3 major conclusions, although there were really 4 to the study. We were scared to do the last conclusion, and you'll understand why in a bit. So the first thing is reproducible changes have been induced by healing intention using the recording. We can do it by direct healing. In other words, put your hands around the cancer cells. We can do it by the recording. And we get essentially the same cancer genes changing, but it's at the very least, we can say we have a technology. Meaning, healing can be captured and it can be released in a maximally scalable system. The question now is it as good as the original? You can see that if it's as good as the original, hands-on or hands around or humans doing the stuff, it becomes much more interesting than an individual healer because it becomes a globally scalable phenomenon. So the second conclusion is we have a technology. However, the technology is not developed sufficiently. Meaning, although the recording produces seriously interesting and reliable phenomena, and it produces interesting healings, it's not as strong as hands-on. So we have the potential of a scalable system. We have the potential for a technology, but we don't think we've captured everything. We don't know why yet we haven't captured everything. So the good news is we have a technology that we've demonstrated that can be worked on, can be made better. The bad news is we haven't arrived yet. So think of it as the Model T. We've got the Model T of scalable healing. This is going to be very different than a modern car in 2022. Modern car in 2022, have more bells and whistles, is going to be, frankly, better. Our healing in the recording is not yet, frankly, better. It's less, even though it does stuff that's interesting and reliable. And so we published these three. We didn't publish the fourth thing that we discovered because we figured it was too much for biologists, and that is when we play the recording in a place, it changes the space of the place. And so we have changed an area into a conditioned space. And the conditioned space lingers the healing effect even after the recording has been turned off. I suspect that will also be the case in human healings when you got a human healer and a human healee. But what we have, I'll give you a quick down and dirty experiment. We call the lab, we tell them to run the recording in an empty incubator. And then you turn off the recording, unhook everything, nothing's going on. We wait 3 days, and then we stick into an inert incubator the same cancer cells, and the genomic changes still occur. So something has happened. Now, for those of you who are basic scientists, think of the methodological implications of this fourth conclusion. If what you're doing in an incubator stays in an incubator, if what you're doing in a room stays in a room, if you're doing in a lab what stays in a lab, and then you come along and you do an extra experiment at the end, think of the mistaken interpretations of data that you might have. You think you've got an empty incubator, so nothing's going on, but in the incubator, there's the residual effect of the recording. So whatever the recording does, you're not looking at a pure experiment anymore. You're looking at something which is interactive with the recording. And you don't even know it. We were too scared to publish this. Just call me chicken. I'll say it out loud, but I'll deny it if I'm on the street. So the conclusions just in the recording are, this is interesting. Something's going on. It shows potential. If you might be able to get there, maybe we can make it better than a human. And I'll get to that when we get to water. So the question was, we can play this recording to cells. What if we play this recording to mice? Will we see the ulcerations? Will we see this? Will we see that? This is also at Brown University. Except, and so now we have the speakers not inside an incubator, but they're just in a mouse room. We've got the same amplifier, and when we play the recording, these are cages filled with mice, and we see what happens to the mice. And what happens to the mice is they start to go into a similar pattern as the treated mice. It's not as strong, and we ran into a complication at Brown. The animal ethics people came in and said, you can't have mice that look like this, even though we told them it was going to look like this. And they came in and they killed the mice. So we lost the ultimate data, what was going to go on, even though the mice, they acknowledge, showed no signs of stress, illness, bright-eyed, bushy-tailed, having a good time, even though they had these ugly tumors hanging out the side of their mouths, outside of their bodies. This too became an interesting traditional biology paper. And we could publish a bunch of things. We can show biological effects in the mice. Certain kinds of cells are affected. The blood is affected. The organs are affected. And some of this is not subtle. So for example, spleen weight increased 5 times. That's an indirect sign of immune function. And something is going on here. And it's not subtle. Something goes on with the spleen, something goes on with the white blood cells, something's going on, but we couldn't carry it to the end. Spent a couple of years trying to get another lab going to try to get this, and finally found one. Actually, I didn't, but one of the SSE folk, who I'll introduce you to in a second, found this at Tokyo University. And here in Tokyo, we got mice and we tried the recording with breast cancer cells, with bladder cancers, with melanoma cells, all injected into the animals and exposed to the recording and to see what happens. And this is, Paul is the SSE Japan representative, and he gets the kudos for finding the lab and being an integral part of the experiments. And essentially we did the same thing we did at Brown. We set up with the exact same speakers and wires and everything else, but essentially the same amplifier. And we found some reasonably interesting things. In the breast cancer model, this is the recording played to breast cancer, or mice with breast cancer, we had very significant effects. The tumor is smaller. Everything about the mouse is better. Difference in tumor volume, difference in mass, difference all over the place. The recording did not affect melanomas. We have no idea why. So the breast cancer, in the breast cancer model, we have significant effects. In the melanoma model, we don't have significant effects. So it's not a universal thing for all types. It's working on this more than it's working on that. And we're just starting this out, so we don't know how far this goes. The bladder cancer, I think we had corrupted cells because they never developed tumors. We tried the experiment a couple of times, and we couldn't seem to get the thing to go in the bladder cancer. So short version, breast cancer model, recording on mice works. Breast cancer model, recording on melanomas, doesn't work, meaning no significant result. And we have corrupted cells in the bladder cancer. But you can see we can take this stuff, we can take these experiments on the road. Turns into a pretty interesting paper. And let's move on now to different form. So all of those were about what happens if we record. And the reason we're going to recording, again, first, maximum scalability. Now let's look at water, which we've already seen. We can treat water and feed the water to cancerous mice. It'll cure those mice. And we got a bunch of clinical data to that effect too. But that simply means the healing can be stored in water. The question is, can it be scaled, and is it as good as the real live treated water? So again, storability, scalability. So the question I have here for the inorganic storage of healing water, is there any way to enhance treated water? And we played around with a bunch of things, and we found out with one, we were talking to a bunch of homeopaths. Now, I'm not a homeopath, but we were talking to a bunch of homeopaths who were talking to me about dilution and succussion. Succussion just means banging something, agitating it greatly. And in homeopathy, again, I'm not a homeopath, we're taking substances and we're diluting and we're succussing and we're diluting and we're succussing and we're diluting and succussing. And in homeopathy, contrary to conventional thinking, current conventional thinking, the more dilution you have, the stronger it gets, with succussion in between each dilution. And we wondered, what would happen if we tried this with our water? So prop number 2, let's start with water. So starting with water, and this is literally what we did, different container, but we started with water and we treated the water. Just as we started with cotton to see what would happen and treated the cotton. We know cotton can store healing. We don't know how to scale cotton. We know that water can store healing. Homeopaths think they have a method using dilution and succussion to mass produce it and to make it stronger. So we started to play around, and you got to understand, this seemed like craziness, to be kind, to me, because I'm taking water, I'm treating the water, and then I'm taking the water and I'm diluting the water. I'm diluting the water with water. Now, what in heaven's name does that mean? And then in between diluting it with water, I'm pounding the thing on wooden benches and all sorts of stuff. And so we're pounding, we're succussing, we're diluting water with water. We're doing this, we're doing that, because we know water can store. But what if you dilute water with water and you succuss it? I mean, this is crazy talk. Except it seems to make the water stronger. And I know that doesn't make any sense. And I'm reporting without comprehension. Again, I don't have a good theory. And we tried different combinations of dilution and succussion. How many times do you dilute? And we did a boatload of them. And we did how many times do you succuss? And we did a boatload of succussions. And I worked with some intuitives, say, help, this could become too complex a problem. Help me out. And so the intuitives would intuit, and we dilute and succuss according to their intuition. And the skeptic in me has to sit here and say, I think they were right. I think they were right. They made it better. diluting and succussing treated water with water that has not been treated, I don't know what I'm saying. It makes it stronger. And so we settled on a couple of formulations for different reasons, and I'll show you some of this. We settled on a promising formulation, which we called Restore. And Restore is treated water that's been diluted and succussed a zillion times. Now again, in homeopathy, homeopaths think, I'm not putting it down, homeopaths think they have a technology. You can take a small amount of a diluted substance and make a boatload of diluted substance. In other words, it's scalable. I came out with a plan B. I invented a gizmo. Gizmo is a technical word. So I invented a gizmo that would do the preparation that, to an extent that you could plug in some water out of a faucet, put it through the gizmo, and out would come the equivalent of diluted and succussed water, which would make it scalable. So I have a technology to reproduce and scale water, and we have a couple of dilutions and succussions. One is Restore, which we use for general health. One is Perform, which we actually use for athletic performance. And one is Recover, which we actually use as an antiviral. And more to follow. And we've actually bottled this, and this I got to go over quickly because this is not a sales technique. So we actually have stuff called Perform, Restore, and Recover with more to follow. Now what we did, because I got to test all this, is I did 3 pilot clinical studies of about 100 volunteers each with Restore, the diluted and succussed treated water that came out of a gizmo that can be mass produced with essentially unlimited amounts of treated water. You don't need a person sitting there doing it. You don't need fancy things going on. This seems to reproduce the treated water. So we did 3 clinical trials of about 100 people each. Everybody had to come in with some serious thing. It could be a physical problem, it could be a mental problem, it could be spiritual malaise, it could be, but you had to be in trouble. And so we had, in 300 people, 2 of these were done in the States, 1 was done in Europe. In the 300 people, we had a lot of different conditions. And some of them were physical, some of them were mental, some of them. And the volunteers took 2 drops of Restore 4 times a day, sublingually for 8 weeks. And we checked in on them every 2 weeks, and they self-reported how they were doing. And this drives me crazy because this is what happened in all 3 clinical trials. 98% of the volunteers reported some improvement or other. Only 2% did not. Virtually everybody wants more. Again, it's not a sales pitch. Virtually everybody wants more. And we had, and I got statistical data and all that kind of stuff. Significant changes in their physical condition, their mental condition, and their spiritual conditions. We had people who were hospitalized with bipolar on heavy meds, leave hospitals, don't need their meds anymore. I mean, the stuff wasn't subtle. Drives a skeptic like me crazy. Wide variety of conditions are reporting changes. We got cancer data, arthritis data, depression, anxiety, pain, Parkinson's, and I can keep going, but you get the plot. And some of the things, like Parkinson's, I think they're stronger than the hands-on treatment. Before we were comparing hands-on to the recording, I don't think the recording's as strong as hands-on. I think this treated water is stronger. I know that's crazy, but I think it's stronger. So for example, Parkinson's case, this is an N of 1, it's an anecdote. In my healing system, we're not particularly good at stimulating things which are missing. We're better at removing things that you don't want. So as an example of removing things you don't want, if you've got Alzheimer's, you got junk on the brain you don't want the junk on the brain. If we treat, the stuff comes off the brain. If you've got a malignancy, you don't want the malignancy, can we get rid of the malignancy? We're good at that kind of stuff. And depression, anxiety, and pain, and all those kind of things. In the case of things which are missing, in Parkinson's, you're missing something that the brain's not putting out. In diabetes type 1, you're missing something that the brain's not putting out. We're not very good. So we've had some spectacular failures with some clinically with conditions such as Parkinson's. And this is an anecdote of one. Some guy in a wheelchair for 5 years, hadn't been out of the wheelchair in essence for 5 years, taken the Restore, threw out the wheelchair, threw out the walker, and is walking a mile at a time. I don't know what to tell you. We've never had that kind of result with hands-on. We're getting it with the water. You see the difference between this and the recording. The recording is more scalable, but it doesn't seem to be up technologically to a live healer. The water seems to be at least as good, but it's not as scalable as the recording, but it is scalable with my little gizmo. And we've done this with different combinations of dilution and succussion. Perform, we don't have as many systematic studies, but we've given this to people running marathons, cyclists, weightlifters, athletes of various stripes, and they put a couple of drops of Perform into a glass of water that they're getting out of the tap, and they're alleging, I don't know what to tell you, they're alleging the weightlifters are breaking their personal record by a lot. The cyclists are going faster and longer. The strength is higher, the recovery is quicker, the oxygen intake, some people have measured that, it's changing. This water is doing something and it's scalable. It's reasonably interesting. We've taken the water, a slightly different variation, none of the three that I've mentioned here, and we've done experiments on plants. And so this is primarily being done in Europe, and we've got factories raising tulips, and we try putting some drops of water into the water system of the plant, and the plants are changing. The tulips are changing. The mass of the plant is changing. The physical structure of the plant is changing. Something's happening. I don't think the plants are believers, and we're putting just a couple of drops of water into the water system. And this is a reliable phenomenon. Reasonably interesting. I was hoping to have reported to you a study on COVID, and I'm going to report to you a half study on COVID. And so this is slightly outdated. I just got data dumped yesterday, so I can't, we're doing a study just clinically and anecdotally. We've observed that we can take some of this water and we can give it to people, and it seems to get rid of COVID very, very fast. But we got to do that in a formal kind of study. And so what we just had done is a clinical experiment on COVID, and we had not people who I tested positive. They not only tested positive, they were about to enter a hospital because it wasn't a casual thing. These are people hospitalized, getting them on the way into the hospital, and then they would be given two of one the antiviral and one the Restore, one to promote health and one to go after the virus. This was supposed to be, and you can see that it's not what I think. It didn't turn out. We're not done. We were supposed to do this in 2 stages. Stage 1, historical data. We did this, 100 people coming in. We didn't give them anything. They just stayed in the hospital. I don't know yet how many of them lived, but they were just in a hospital, and we did whatever we did. The next week, we started what I had hoped was a double-blind, randomized, placebo-controlled trial of the next 200 people entering the hospital. They were to take the charged water and to take an uncharged water as controls. So the volunteers in stage 2 were given 2 formulations taken sublingually for 7 days, one inform, one perform. So I come here and I say, it wasn't done right. The background study showed that that we got that. We got 100 as a historical baseline, but the randomization of the groups that were getting the water versus placebo, in the administration of this, some of the administrators, and these were physicians, mixed up the bottles, and not everybody got the right bottle that they were supposed to get. And so I don't have all the data yet. We're still missing some of the respondents, but all I can tell you just as a tease is this. And I just looked quickly at the 7th day. On the 7th day after being admitted to a hospital, looking at general how do you feel, there's a seriously, seriously significant difference between the people who got nothing in the historical study versus the people who took our drops. I mean, it's really serious. But I can't say that yet. It's not a double-blind, placebo-controlled, it doesn't have the gold standard and all that kind of stuff. But I just say it's discouraging, but the preliminary stuff is encouraging. But I can't give you the results. And maybe I will by the time of the conference. So if we get it to the Q&A, with luck, I got another week to get this done. I'll have more. So if you ask me about this, I'll tell you if I have anything else. So how do we pull this together? Well, again, scalable but not as strong recording isn't as good as live, but we can give it out. Water, which seems to be stronger but is less scalable, but now I think I can make it scalable, probably has more. Doesn't mean we should stop working on the recording, but in the short run, the water probably has more oomph. Let's do this as a thought experiment. Let's assume, just for fun, now this is not the case, this is a thought experiment. Let's say that I get the data from the COVID study, and let's say that what seems to be the case really is the case, and that is the drops get rid of COVID quickly. And we have over 300 people that have been given this thing outside of experimental conditions. And it's pretty obvious that something happens. But under controlled, placebo, double blind, all that stuff, let's assume that it still happened. Let's assume that we then have a viable treatment. I don't know if you're going to call it a cure. We don't know enough. But we have a viable treatment for COVID that gets rid of COVID, and it's just water. Now, let that sink in. That's unconventional. It's just water. What would I do next? Would I stand up and say, I've got a bunch of water that'll fix COVID? Now, who would buy into that? And I'd say to the basic scientists, I got a double-blind, placebo-controlled, randomized, yada, 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 yada. And they'd go, okay, do it again, do it again, do it again, do it again. I don't believe you. Do it again. I don't believe you. Do it again. But a normal person who had COVID would say, I don't care about those questions. Give me the damn drops. People all over the place should want the drops. They don't have side effects. Can't overdose. It's water, for crying out loud. But it fixes COVID. What if we have water that fixes Parkinson's? What if we have water that fixes, you get the idea. So what if we come up with water that does all this? What's the next step? So here's our problem of converting from the unconventional to the conventional. And if the unconventional comes up with a reliable system that conventionally can fix measurably these conditions, a normal layperson says, I don't care, give me it. In the same way, incidentally, that our plant experiments going on in Europe where I say, this doesn't make any sense. I'm putting a couple of drops into the water system. I can't think of the reason this would work. But the owner of the factories say, we don't care. Our tulips are better. So the basic scientist is going to say, yeah, but. And they say, give me the stuff. I want good tulips. I come along and say, to the basic scientist, I have water that'll fix COVID, or I have water that'll do Alzheimer's, or I have water and they go, get out of here. Do it one more time. Think Pear Lab. One more time. Do it for 30 years and then come back and talk. But the person who needs it, The person with Parkinson's, the person with cancer, the person with Alzheimer's, the person with is going to come along and say, gimme. I said, but I don't know what it is. Gimme. And think about the person who takes this, gets fixed to COVID, might change their life. Might. Who knows? So what happens, I'm not sure where the possibilities can keep going. We said the recording conditions space. What if we condition space in a medical facility? What if we condition space in a hospital? What if we condition space in a medical clinic? What happens if we supply clients with treated water? What happens if we supply them with treated water in a place that's already been conditioned? Can the recording, can the drops enhance clinical outcomes, experimental outcomes, plant outcomes, animal? I got stuff on animals too. You see where this might become widespread, and you can see how the different mindset. I make a claim, consciousness can affect random number generators. Give me 30 years of data, 30 years later. Give me 30 more years. I come in and say, I can fix COVID. 30 years I want, says the basic scientist. The COVID patient says, gimme. That's what I'm talking about. A lot of our stuff can be harnessed, changed, modified, developed into a technology that'll make it more conventional. There's all sorts of studies to be done there. So special thanks to the folks who have done this: Brown University, Institute of Noetic Sciences, Tokyo University, Beech Tree Labs, generous funding from the Emerald Gate Charitable Trust. And I'll leave you with my favorite cartoon. I think it's a copyright violation. It says, when Dr. Bengston comes in, everybody play dead. Anyway, thanks for listening. Q&A. Maybe I'll have more COVID data for you. Who knows? And I hope you're having a good conference.